I'm going to be reading the questions that were submitted online. The website was open for questions through, I think, about 4 p.m. this afternoon. If there are questions that have arisen through the presentation or as we discuss these questions, there are additional cards at the back table that you can submit that. Again, if you include your contact information, if you did not get your specific question answered in all the detail that you provided or were hoping for, you will get a response via email if you provide that information, and the website will be updated with these responses. So with that, the first question that we have is, how will these genetically engineered moths affect humans or other animals that might ingest them?
I think Tony kind of touched on this in his talk. So we've developed more than one such self-limiting insect, as well as a diamondback moth. These insects all express the same protein, the self-limiting gene and the marker gene. They've been subject to independent assessment of their hypothetical toxicity to off-target species, predators, parasitoids, with no evidence in these tests of any ill effects on the non-target organisms tested. This and other data has been, assessed by independent experts at the USDA, who also concluded, as part of their finding of no significant impact, that these insects are non-hazardous to, people or animals that might ingest them.
Okay, I think this might be kind of a follow-up. Someone wanted to know about the impact of the release of the moths on, specifically on bees, and whether or not this would potentially contribute to their decline.
So what's really interesting about the sterile insect technique and this self-limiting is that it's species-specific. So as species-specific, it doesn't, it just goes after other diamondback moths. So if you don't spray for diamondback moth, if you control it through this biorational method, then you're going to be reducing the pressure on the pollinators and the bee population, because you really don't need to control diamondback moth with insecticides. So all insecticides, most all insecticides, have a breath to them. If you apply something to control one insect, it may also control or do harm to another insect, one that you want to preserve. So I think that's the beauty of this self-limiting, or as was the sterile insect technique, is that it's very species-specific.
Okay. There's a question about how the release of the genetically engineered moths will affect organic farms.
Sure. So organic standards say that you cannot use any synthetics. You can't use synthetic insecticides, you can't use anything genetic engineered or not. So organic growers would not be allowed to use this technique with their existing regulations. I think that would be a little bit disappointing, though, if we see that something like this technique really, really works well, and some farmers are not allowed to use it. I think that's a difficult situation. I think one of the other parts of this question is what happens if a moth from our project flies over into an organic field. Well, again, in our project, in our specific project, we have a very, very isolated field. And if we're evaluating just our project in this, the likelihood that an insect would travel from our research field, 5 miles away to their farm, and that they would be able to even find it is minuscule. Organic farmers have also have experience with genetically engineered organisms, becoming present in their field, like, for example, with genetically engineered corn. And if the pollen lands in their fields, and all the legal cases that I'm aware of, and I've talked with a number of people who specialize in this area, say that their farms would not be decertified because of this adventitious presence.
If farmers have cabbage and other brassica fields near the Geneva test plots, will those crops be affected by the insect releases at the experiment station?
Well, again, I think it's the same answer. We are working in a very, very isolated research part on the farm, and I can't see exactly how the insects would move over into an organic farm.
Okay. Now, that was not specific to organic farms, but I think the answer might be the same. Is there a concern about non-organic farm
It
proximity?
would be the same answer.
Okay. Okay. Since the diamondback moths generally travel on the wind, how can you be sure that they won't leave the site on a wind gust or a storm? Such things are frequent in our area.
Okay. Again, the conditions for the release that were approved by BRS, if there is a potential storm coming, we would not be doing any releases. So that would negate largely, I think, that concern. Also, we are in such an isolated area, surrounded by woods. Wind normally comes from the west. We have woods on the west, we have woods on the east, we have woods on the north, or on the south. Could it be blown over there? It would be very, very difficult, very unlikely.
Have you sufficiently assessed the human health risks associated with this technology?
I guess this leads on from my previous response in that the US Department of Agriculture, as part of the process that Tony described, assessed safety to human health and the environment. That was essentially the scope of their assessment of these trials. And the result, finding no significant impact, arose from, for example, assessment of the non-toxicity and non-allergenicity of Diamondback moths. I guess in a nutshell, these moths are non-hazardous. But to take that a step further, this is a very small trial relative to much larger scale agricultural trials. So in the context of the wider agricultural landscape, as Tony suggested, the trial site is very located, and actually the chance of these moths coming into contact with people is very small indeed. Tony, I don't know whether...
So I'd say any hazard of these self-limiting moths would be if consumed, it would be the same thing as a non-engineered moth and consuming that. If the two proteins are safe and non-allergenic, then I think that the any potential hazard would be the same. And, you know, those of us who work in entomology, and we've over the years consumed moths, not as a steady diet, but it's something that, you know, we don't really fear. You know, there's a whole science of entomophagy about eating insects, and 80% of the world's population eats insects. If I eat a diamondback moth, I'm not going to grow stronger, but maybe I will.
Okay, these two questions are somewhat related, so I'll read them together. Can you explain specifically how the insects are developed and what makes them self-limiting, and how this genetic manipulation is different from a Roundup-ready technology?
So I can certainly take the first part of that and see how we go. So in developing these moths, we've essentially started with a colony of non-modified diamondback moths in the lab, and we've collected the eggs of these moths, and using a very fine needle, we've injected synthetic DNA into the eggs at a very early stage, and with specific sequences, that has inserted into the genome of the diamondback moth. And with the use of the marker protein that Tony described, we're able to then, in the next generation, identify individuals that carry this self-limiting gene as a heritable gene. So it's passed from parent to offspring and becomes heritable just like any other gene in the genome. So the self-limiting aspect of that gene really arises from a genetic system called the TET-off system. I won't go into too much technical detail, but its mode of action isn't through a toxic effect. We've utilized this system in combination with a moth regulatory sequence that essentially drives gene expression in females only. So we can ensure that this genetic switch only turns on in females, and the end result is that essentially gene expression of this specific gene becomes so high that the machinery used by other genes to produce proteins that are necessary for normal function of the cell isn't available, and so those essential genes are starved and not able to produce those essential proteins. And so the cell dies and the insect dies. And the genetic switch, so we've made that female specific. So in the diamondback moth, it prevents females from developing to the reproductive adult stage. And so when we release our males into the field, their female offspring do not survive, and as Tony described, with sustained releases of these males, the population shrinks. In the factory, we're able to turn this switch off. So if you're trying to produce large numbers of moths in a factory-type setting, it's nice if you can have males and females. And so we add a chemical to the feed of the juvenile diamondback moth, which switches this gene off. And so males and females are able to survive and we can reproduce as normal. So if I can kind of sum that up, it provides a way of being able to produce moths as normal in a factory, but when we want to produce male-only cohorts of moths to release into the field, we can do that by withholding the chemical from the feed of the larvae. And after these males are released into the field, they result in a crash of the number of female diamondback moth in the pest population.
Okay.
Might add one other comment to there. So most other insecticides, you know, whether it be a pyrethroid or pyrethrum or neonicotinoid, they have certain target sites that toxin will attack and disrupt, say, the nervous system. Most of the most of the insecticides we have are those nervous system toxins. This is totally different. This just affects the reproductive ability of the insect. And when we compare technologies, it's important, although we're focusing on this experiment, which is on reproductive disruption, you have to compare any risks and benefits of that to the risks and benefits of any other conventional insecticide. I think it's always wise to put those comparisons out there.
Okay, these two questions I think can be taken together. Previous trials have shown 1% or more survival of the genetically engineered larvae to adulthood. What is the expected life of the efficacy of the genetically engineered diamondback moth as a control strategy? That's the first question, but related to that, it says that in studies for mosquitoes that were developed by Oxitec, late lethality was an advantage for controlling insects like mosquitoes, but would not be the best strategy for insects with an essentially infinite food supply like agronomic pest insects. Given this, why is late lethality an appropriate strategy for the diamondback moth? So I'm coupling those two together because they sound to me like they might be related. If they're not, please feel free to uncouple those two.
Can you repeat the first one?
Yes. So the first one said that if previous trials have shown 1% or more survival of the larvae to adulthood, what is the expected life of the efficacy of the genetically engineered diamondback moths as the control strategy? I think the question is asking.
Yeah.
One question.
No, that makes sense to me.
Okay.
So early published papers described very small numbers of females surviving to reproductive age. But actually, in far larger tests and much more controlled tests, we now see no females surviving to reproductive age. But I think the question essentially alludes to what's the likelihood of resistance developing to this approach. And I think it's fair to say that any pest management approach is no pest management approach is immune to resistance developing to it. However, we've released over 200 million of our mosquitoes around the world, self-limiting mosquitoes, with no evidence of resistance arising despite these very large-scale trials and operations. I think, our aim, certainly with the diamondback moth, is to provide one tool in the toolbox to farmers as part of an integrated pest management solution. And I think that approach is one step towards mitigating the potential for resistance to develop to any pest management tool. If you reduce over-reliance on one single tool, you reduce the pressure, the selection pressure for the pest to develop resistance to it. And so, while we don't have any evidence of resistance to developing to our approach, I think you can certainly take technical steps to mitigate that hypothetical risk.
And then I apologize for combining those two.
Okay, so the second question, I believe, was is late lethality, and by that I assume that what's being meant here is lethality in the late larval stages, right? An appropriate strategy. So if you're thinking of this using a chemical pesticide paradigm, no, that would not be a particularly good strategy. But if you're thinking of this as a component of that avoidance base that I portrayed in that pyramid, yes, it is. Because you're not necessarily trying to drive an existing high population down and thereby prevent damage. Really what you're doing is you're using tactics to never achieve a high population to begin with. So you have multiple generations to do that, and you rely on several of other of those processes to help you achieve that goal.
Okay, there are two questions about logistics. So in the original permit to USDA, which was resubmitted, you were also permitted to release non-genetically engineered moths if there was an insufficient natural population of the pest. Will you be releasing, excuse me, or considering releasing non-wild, non-genetically engineered moths as well in this round of open-air experiments?
Yes, and what we are doing is comparing the behavior of the non-genetically engineered insect to the genetically engineered one. So yes, that is the comparison.
I think just to add to that, the intention of these releases is not to infest a field with a pest, as I think the question is alluding to, but as Tony suggests, compare the behavior of male moths together.
Okay, and what do you believe is gained through an open-air trial as opposed to doing a trial in a 3-season, multi-acre hay grove structure, which would ensure that the moths do not escape?
In a, what was that, a 3-season?
3-season, multi-acre hay grove structure.
Well, so many of the farmers in this area, when they have a field of cabbage, it might be 30, 40 acres. So we need to understand its biology under those conditions. That is its movement patterns and its ability to find and mate with insects, and that was what I was describing with what we call the sentinel female experiment, where we release the males as 2 types of males, and we have them seek out these females and then mate with them, and then we assess the offspring. So yes, we do need these larger experiments, but again, this is done in an isolated field. So.
Okay. Have we studied the long-term effects of this technology and the genetically engineered moth over the next several generations?
Yeah, I could start by potentially answering one element of that, in that we've certainly held this strain of self-limiting moth in the lab for many years, but also many generations. And part of that process has been to periodically assess different properties of the moth and check and verify that its properties remain the same over generations. And nothing unexpected happens with regards, some of its basic biological properties. We've also looked at, single experiments over several generations. So, in lab experiments, we've looked at, we've essentially simulated the persistence or lack of persistence of self-limiting moths in populations of Diamondback moth, and found that after you stop releasing your moth into a contained population, the gene rapidly disappears from this population in a small number of generations. And that empirical experimental result closely matched that of mathematical modeling, which predicted exactly the same pattern. I've answered maybe 2 sides of that question. I don't know if...
I think that answers the question. We now have a series of questions that are more generally related to genetic engineering. Is this the first release of genetically engineered animals in the United States?
Well, I'd preface my remarks by saying that there have been billions and billions of modified insects that have been released in the sterile insect technique programs.
And I'll follow that maybe perhaps by quickly describing previous releases in the US of of another moth that was genetically engineered, this time with just the marker gene that we're talking about here with Diamondback Moth. So with the US Department of Agriculture, we developed a genetically engineered pink bollworm, which is a big pest of cotton in many parts of the world, including southwestern US. So they have long had a sterile insect technique program running against the pink bollworm, and including a genetic marker that was visible, as described for our Diamondback moth, was attractive to them as it offered them extremely reliable monitoring and consequently much more efficient program, I guess, efficiencies. So we developed that moth with the US Department of Agriculture, and since 2006, they have released over 20 million moths on cotton in Arizona as part of that sterile insect technique program. It just so happens that transgenic BT cotton came along at the same time, and in combination with the sterile insect technique, they have successfully eradicated pink bollworm from the entire southwestern USA. Of course, that means they don't need to release this genetically engineered moths, so those releases stopped back in 2015.
Okay. Are there plans to apply this technology to other animals? I think we understand you can only answer for yourselves.
Yeah, I want to give everyone else a chance as well. But at a later stage of development is our self-limiting mosquito. It's an Aedes aegypti, which is the mosquito responsible for the principal vector of dengue fever and Zika, and yellow fever, and another viral disease called chikungunya. So these diseases, they affect hundreds of millions of people a year, cause debilitating symptoms, and as we know, also death. So we've developed self-limiting Aedes aegypti mosquitoes. These have undergone trials in Central America, the Caribbean, and Brazil. And all of these trials have produced results in which the level of mosquito control far exceeded what's possible by conventional methods. And so, it certainly demonstrated that what we have is a very powerful way of controlling this difficult to control mosquito. In Brazil, we've moved a step closer, actually. So we now have a factory near Sao Paulo that has a capability of producing 60 million of these self-limiting male mosquitoes every week. And over the coming years, that's enough to potentially treat an area of over 3 million people. So, at that scale, obviously, we're at a much more advanced stage than what we're doing with agriculture. And beyond Diamondback moth in agriculture, we're also working with some other important pests, such as the Mediterranean fruit fly, which is one of the world's biggest pests of fruit, and also the spotted-winged Drosophila, which Jan described, invaded the US, North America, Europe over recent years, and is extremely difficult for growers to control. So in parts of Europe, in berry and soft fruit control, they had got to the stage where they could produce soft fruit without the need for any insecticides, really relying on biocontrol and other methods. This fly came along, and it was back to sprayed insecticides, and they were back to square one. And so we're in the process of developing a product that we believe can help to remedy this problem in many parts of the world.
So, also, there are researchers at universities like Max Scott down at North Carolina State who have genetically engineered screwworm. This is the one that is controlled by radiation, but it's not really there's got to be some improvement. And so he's been able to genetically engineer a screwworm, and I think he got a permit to do some releases in Panama. He's also working on spotted wing Drosophila. There are other scientists at other land-grant universities working with genetic engineering of insects as a tool. So, with all due respect to my colleague at Oxitec, there are some other players in the game. And many people, many researchers see this as a tactic worth exploring more because it has some tremendous possibility. There's someone from MIT who's trying to control Lyme disease through some genetic engineering. That's a little bit different tactic because he's actually trying to engineer the mice, which are one of the hosts. But, we have to ask ourselves in, 5, 10, 15, 20 years, is it really going to be responsible for us to continue on this heavy insecticide-dominant way of controlling our pests? There's got to be other ways. And we, as Jan was talking about that in that diagram, we all work on biological control. We work on host plant resistance. We work on landscape management. We work on many, many different tactics as part of an IPM program. I think it's important that we also look at these self-limiting insects.
Okay, this person writes, I've read some articles about the technology called CRISPR and another technology called gene drive. Do these moths use either one of those technologies?
I could start and see if anyone else wants to chip in, but no, our approach doesn't use CRISPR. So CRISPR is a really exciting technology with potential for, so technology used for gene editing. So it has tremendous potential in some fields, not least to solve inherited genetic diseases of people. Another potential application is the second half of the question, gene drives. So gene drive is quite a hot topic in research and would essentially involve the release of, let's say, insects, mosquitoes, carrying a gene that is able then to spread through a population and change the genetics of a population, potentially to the benefit of people. One idea is to change the genetics of populations of mosquito to make them genetically incapable of transmitting disease. Our approach is different, so it doesn't use CRISPR, but it also does, in one way, maybe the opposite of what gene drive does. It doesn't replace populations. It's a self-limiting gene that after you release it, after you stop releasing it, the gene disappears from the population. We're trying to reduce pest populations rather than replace them with another version of the pest with different genetics.
Okay, this one is getting back to the specifics of the Diamondback moth. In New York State, the Diamondback moth is often managed jointly with imported cabbage worm and loopers. Given that many farms spray for these three pests combined, how will the use of genetically engineered Diamondback moths reduce pesticide use in the field?
Yeah, it's a good, interesting question. Imported cabbage worm is certainly, it's an endemic pest that we have here. It's the only caterpillar on cabbage that is endemic to this area. There's been a long use of releasing biocontrol agents for imported cabbage worm, and any grower who's been in business for a while has seen remarkable declines in the imported cabbage worm population due to this biological control. Cabbage looper is another insect pest of cabbage. it's a migrant up. And we're seeing it much, much later in the year now, if we see it at all. But we continue to see diamondback moth. The insecticides that could be used, say, for imported cabbage worm, if you whisper insecticide to imported cabbage worm, it'll fall over and die. It's just a very, very susceptible insect. Diamondback moth is not that. So if you can control diamondback moth through the use of, say, self-limiting technology, I think you're much, much better off, because biological control is really working to reduce the imported cabbage worm populations throughout New York State. There was a large USDA project, we were involved in it, to bring in a new parasitoid, and that's worked very effectively. That kind of biological control is not really suitable for diamondback moth in this area. In other areas it might be, but because diamondback doesn't overwinter in this area, the parasitoids are going to have a long lag time before they become effective.
So I just want to add one comment to that. And it's important that we remember or continue to keep in mind, I mean, by the way, the question is a good question and ultimately an important question. But what we're working to do right now is to answer a couple of questions, biological questions about this technology, right? And that, at this point, we are not saying how necessarily is this technology going to integrate ultimately into an integrated pest management system. We are doing technology assessment about this particular approach. Eventually, we would have to answer that, but we have to do this assessment first.
Thank you. This question asks, have you evaluated whether or not the tetracycline switch that you will be using will add to antibiotic resistance within humans, insects, and farm animals?
I guess there's two sides to this. I think, taking literally the actual switch itself, the TET-off switch, which responds to tetracycline, which is the antidote used to turn off the self-limiting gene. Components of those genes are not, they can't be passed on to other organisms other than diamondback moth, and in the context of the self-limiting insect, are only useful for controlling a pest population in the field. I think another side of the question is that to maintain populations of self-limiting insects in the lab or the factory, you need to provide tetracycline or a suitable analog of tetracycline in the feed of the juvenile stages of the insect in order to switch that self-limiting gene off and allow, in the case of diamondback moth, females to survive. So with our diamondback moth, in the generation before release, we don't provide them with tetracycline. And so they're reared in the absence of tetracycline. And so in that generation, they're tetracycline free and resistance isn't being selected in that generation. I think, on top of that, this is a small trial. These are very small moths, and the biomass of bacteria in these moths is extremely small. Tetracycline resistance is common in soil bacteria all around us, and so, I think factors like these were assessed by the US Department of Agriculture as part of their environmental assessment, which resulted in the finding of no significant impact on a collection of issues, including this tetracycline resistance one.
Okay, and also referring to tetracycline, if that is present in the environment, will it turn off the female lethality trait switch, allowing significantly more of the females to survive?
So, again, this was all part of the US Department of Agriculture's assessment, but if I can summarize some of that thinking, tetracycline out there in the environment, in the elements, under the sun or in the soil, is unstable, so it doesn't persist for very long at all. On top of that, perhaps the most likely source of tetracycline might be from farm manure, from tetracycline-treated farm livestock. And untreated manure is not commonly used to fertilize commercial fields of cabbage. Moreover, brassica crops, cabbage, not very efficient at taking tetracycline up. So I think there's a number of steps in which add up to the likelihood of sufficient levels of tetracycline reaching a self-limiting insect on the crop. Is highly unlikely to be present in sufficient concentrations to rescue that self-limiting gene.
In our research trial, and it's the only one I can talk about, there has been no animal manure applied to that land for decades. One of the other things looking down the future, down the line a bit, is that cabbage growers I know have really gotten away from using animal manure because of pathogen concerns as part of a food safety strategy.
Okay, I think this question was answered, but I want to ask, it's slightly more specific. So it says the larval form of the diamondback moth is the pest stage for the moth. If these genetically engineered larvae survive and potentially are on other crops, what assurances can Cornell offer that cabbage and other crops that have these larvae will still be salable? I think the answer given previously was that there's not anticipated to be migration to other crops, and that if they were, and they were organic, that would not affect the salability of the crops. But if there's anything additional you wanted to add on this specific point.
Okay. Again, in our trial, the field is extremely isolated. The likelihood of any of those moths leaving our trial and going 5 miles or 10 miles to another field is extremely, extremely unlikely. And again, legal precedents so far have been the adventitious presence of genetically engineered organisms in an organic farm is not a reason for decertifying that farm. And how are you ever going to see or find a small diamondback moth in a commercial farm? I mean, not from our research trials, you're not.
Okay. And the last question that I have, who is providing the funding for the research?
It's very clear that for research that is done, we do need funding. Oxitec is providing funding for this research. However, they have no rights or restrictions on this research at all. That is, we control the data, we control all aspects of the project, and also the publication of the project. That's something that Cornell it's sacred. If Cornell is doing the research, we're a land-grant university, we are not influenced by the funding. If our trials turn out to be, this is just not suitable, I'll be the first one to stand up and say that. But we control the trial, how it's done, the information on it, and that's something that that's just standard practice for Cornell.
Well, on that note, we anticipated limiting this forum to 8:30 so that everybody would be able to get on their way. And we've achieved that objective with the assistance of this great panel. So I would like to thank Dr. Grant, Dr. Morrison, Dr. Shelton, and Dr. Nyrop for putting on the forum, and for everybody who came out and submitted questions.